Hi, I'm John Watt, and welcome to Ever Wondered. This week we're taking a look at one of the biggest scientific challenges we've ever faced: climate change. It seems humanity has become a victim of its own success. Scientific advances in medicine and technology have led to an unprecedented quality of life, but also to huge rises in population and gas emissions. Many of these gases have heat-absorbing properties, so as they increase within our atmosphere, so, it seems, does the temperature, just like in a greenhouse. I'll be meeting the scientists that are using incredible ways to monitor these greenhouse gases and also finding new ways to reduce them. But first I'm off to Wellington to meet a scientist who wanted to find out more about global warming. To do this, she went to the coldest place on Earth. Katja Riedel is looking at ancient Antarctic ice to find out what the future holds for us.
When snow falls, you have a lot of air in it, and over time, when snow gets to ice, you make little bubbles, and these bubbles really contain the ancient atmosphere.
Through ice cores, scientists have already charted greenhouse gas levels as far back as 800,000 years, and shown that they are now much higher than they have ever been. Katja is going to show us how this information is extracted. But first, I have to suit up. Moon boots. Yes. It's definitely one of the weirdest outfits I've worn before. Right.
Good to go.
I feel warm.
Good.
The cold lab is kept at -20 degrees Celsius to protect the samples. It's cold as.
So this is this Antarctic ice core from the Whitehall Glacier.
Oh, so you can see all the bubbles and stuff quite clearly.
Yeah, that's right. And you see the thin lines here and there?
Ah, yeah. What are they?
Melt layers.
Melt layers?
When you're probably in summer or so, when you get slightly higher temperatures, then the surface melts a bit.
Isn't it rad how nature just does it so perfectly for you?
I know.
So how old's this ice?
This is probably around 1,000 years.
By looking at the bubbles, Katja can tell which were formed in winter and which in summer. So the ice core charts the years, much like the growth rings in a tree.
The next important step, get the gas out of this ice. And the way we are doing it is we're actually cracking up all the ice by shaking the hell out of it and open up all the bubbles.
Katja does this with an abrasive device called an ice grater.
What I have here, these are pieces that I've been shaking before, so it comes out like little balls and a lot of snow.
This all happens in an airtight container so that the air released is trapped.
And then we can connect it up here and freeze over all the air into the warm room.
Warm room? That sounds really good. Can we go there?
Yeah, let's go.
In the warm room, they use liquid helium to trap the gases that have been released from the ice. So why do you use liquid helium?
Because it gives us a very cold temperature, 15 Kelvin.
15 Kelvin, that's what? -258 degrees Celsius.
That's right, so it's pretty cold.
Just like water molecules being attracted to a cold window pane, the liquid helium creates a relatively cold surface, which the gases are attracted to. They then freeze in the metal trap. So you're actually using the coldness of the liquid helium to entice the gas from the other room.
That's right.
The gases can then be analysed, but there is even more crucial information trapped in the ice itself, the temperature at the time. This can be discovered because the water contains different oxygen isotopes. These are simply oxygen atoms of different weights. When water evaporates, the lighter water evaporates more readily, but when it is hotter, more of the heavier water can be taken up. This then falls as snow. So by looking at the isotopes preserved in the ice, Katja can figure out what the temperature was when the water evaporated.
It is important if you want to talk about climate change and try to get an idea of what happened in the past to look what the temperature and the greenhouse gas variations did together. So how do greenhouse gases really influence the temperature?
If we look at the carbon dioxide levels of the last 800,000 years and then compare them with the temperature, the overall correlation is striking. However, there are still some inconsistencies, and the more we know about these, the better.
And this might help us to really kind of get an idea of what we are in for. So what will be the impact of now our amazingly high greenhouse gas concentrations on our climate, on our temperature?
Antarctic ice cores have provided us with an amazing ancient history of the main greenhouse gases we produce. These are carbon dioxide, methane, and nitrous oxide. But what about closer to home? I'm heading out to one of the most significant atmospheric monitoring stations in the Southern Hemisphere to have a look at how these gases have been on the increase in New Zealand. Gordon Brailsford is my guide. So where are we now, Gordon?
Well, this is the Bering Head Atmospheric Monitoring Station. It's a place where we can measure greenhouse gases really precisely. We don't have factories or cars right on our doorstep. So here out on the coast, we're away from all those local sources, and the air arriving at Bering Head is not seeing land for maybe 5-10 days.
Bering Head's unique position means that when a southerly is blowing, the air has been crossing nothing but ocean, giving a consistent representation of greenhouse gas levels.
Well, Bering Head's got the longest continuous CO2 measurement program in the Southern Hemisphere, so we've been measuring CO2 at this site since 1972.
So can we have a look at some of your equipment? What's the wood for?
It's to keep the goats out, our local unicorn.
Yeah. It's a fairly busy room. What happens in here?
Yeah, it is a busy room. This is where we make our atmospheric CO2 and oxygen measurements.
Yeah.
So the air comes in from outside, and we
come through there, does it?
Yeah, through that black tube, and then we dry it up and using fridges and freezers, and then make our measurements on the different instruments really precisely.
And so these are some results?
Yeah, this is the last 6 hours of gas measurements from here, and so last night we had a strong southerly, so the air was really stable and wasn't changing very much.
Bering Head has been taking these measurements for almost 40 years and has charted a steep rise of carbon dioxide. This fits with the global trends that show incredible increases since the Industrial Revolution of the 1800s. But Bering Head makes a whole series of other measurements too. Right, so what happens in this room, Gordon?
This is where we use pumps to collect air into flasks and tanks, and then we'll take those back to the laboratory to measure them.
So you're making much more complicated measurements here?
Yeah, so measurements that are difficult to make in the field, we do back in the laboratory in a more controlled environment.
These flask measurements have also charted the dramatic rises of other greenhouse gases, nitrous oxide and methane. But these measurements only take into account the gases that exist near the Earth's surface. To get an accurate picture of the greenhouse gases in our atmosphere, we need to measure from the ground to space. The question is, how do you do it? Here at Lauder in Central Otago, they're looking to the sun for answers. The scientist asking the questions is Vanessa Sherlock.
The next generation of greenhouse gas measurement techniques give you a measurement of the greenhouse gas abundances through the entire depth of the atmosphere.
This is called total column measurement and is much more accurate than previous methods. But why is more accuracy important?
As time goes on, it's increasingly likely that we're going to want to know that nations are in fact reducing emissions in accordance to protocols like Kyoto, and politicians need to know that the measures that they're taking to mitigate greenhouse gas emissions are being effective, and they also need to know that everybody is doing what they signed up to do. We have a very simple telescope which points at the centre of the sun, and we record that sunlight.
From this sunlight, Vanessa can extract some incredible information. The exact concentration of every individual greenhouse gas.
To me, this is part of the magic of the whole thing. Each of these greenhouse gases actually has a kind of unique fingerprint.
This means that every different type of molecule absorbs light in a distinctive way. So Vanessa analyses the light that reaches the Earth's surface by splitting it into its full spectrum. Then she tries to find out what tiny bits of the spectrum are missing.
Okay, so unfortunately it's cloudy today, but usually we'd have the sun coming down from the solar tracker on the roof and being piped into the instrument using this mirror here. And then we focus the beams onto this beam splitter.
This is where the light is split into all of the frequencies of the spectrum and is then sent to a computer for analysis.
You can see at some frequencies there's a lot of signal, and at many frequencies there's no light at all. And what we're really looking at is absorption of sunlight by the carbon dioxide molecule. And each molecule will have its own distinctive pattern, and that's how we know which gas is doing the absorbing.
So is this a very accurate picture of what's there?
These measurements have to be very, very precise and accurate. We've done amazingly well in meeting a 0.2% precision and accuracy goal, but we're trying harder to improve on that.
What do you need to get to?
There's no limit. We can't be too good.
As Vanessa's telescope points towards space, we're looking to a plant that can be seen from space, phytoplankton. Cliff Law is investigating whether they could be used as a way of reducing the greenhouse effect.
Phytoplankton are equivalent to the grass in the ocean, basically. They photosynthesize and capture the sun's energy, and then they actually also use up the carbon dioxide that's dissolved in the water. So that's the carbon dioxide that's moving from the atmosphere, that's just dissolving into the water, and the phytoplankton are drawing that up. When these phytoplankton eventually die, a proportion of them sink into the deep ocean, and they take that carbon with them.
This carbon can be locked away for hundreds of years, but there are large areas of the ocean where phytoplankton don't grow to their full potential. And why this is has been puzzling scientists for years.
This idea came about that they were lacking iron. Now, we all need iron. They need iron for their pigments and enzymes, just like we need iron in our blood cells.
So Cliff went out to an iron-deficient part of the ocean and added some iron oxide.
What did you discover?
We saw a very strong response. The phytoplankton started growing. Effectively, the water turned from clear blue to dark green. We have things like satellite images of our experiments from hundreds of miles above the Earth, where you can see this big green patch covering 1,000 kilometres of growing phytoplankton.
As the phytoplankton grow, they are absorbing huge quantities of carbon.
The idea has been picked up on by various commercial organisations. They're obviously looking at this in a positive way to try and control the atmospheric carbon dioxide, but also they're looking at this as a way of selling carbon credits. Now, there's a big carbon credit market out there at the moment for many different methods, and so they're looking to tap into this kind of thing.
Well, that sounds like a really good way to reduce carbon dioxide.
There's possibilities there, but it's not the panacea it automatically seems to be.
This is because things aren't as simple as they appear on the surface.
When these phytoplankton eventually die, a proportion of the phytoplankton that's sinking through the water column is being broken down by bacteria that actually produce nitrous oxide and methane, which are both strong greenhouse gases. And so overall, you don't know whether you're having a positive or a negative effect.
So companies could be selling carbon credits for something that actually contributes to global warming. And that's what Cliff is currently studying, trying to figure out the overall effect of adding iron to the ocean, so a realistic picture can be painted of its environmental impact.
If there is to be a large-scale release of iron, then obviously this is something that needs to be measured and monitored.
Cliff's expertise is now being sought by international organisations.
Our involvement in this has actually moved from an experimental stage to an advisory stage on policy, where we're feeding in at the level of the International London Convention.
The London Convention is an agreement between all of the countries highlighted in green, which protects the ocean. Cliff is helping to develop the part of the convention that deals with iron fertilisation, so his research is having a global impact. A huge amount of our greenhouse gas emissions come from agriculture. Most of it comes from methane, which makes it a hot topic for greenhouse gas research. Harry Clark is the head of the Agricultural Greenhouse Gas Research Centre in Palmerston North.
New Zealand has a very unusual greenhouse gas profile. If you look across Europe, North America, etc., about 7% of their greenhouse gases come from agriculture.
But in New Zealand, almost half of our emissions come from agriculture, and 32% is from methane.
A dairy cow will be producing around about 90 kilograms of methane per year. If you then say what that is on a carbon dioxide equivalent basis, methane has a value of 21, basically meaning it's 21 times more potent than carbon dioxide. So you come to 2 tonnes per animal per year.
To put that in perspective, just one cow emits the carbon dioxide equivalent of around 860 litres of petrol per year. That would power an average Kiwi car for more than 7 months.
In New Zealand, if we just want to reduce emissions, it's very, very simple. Get rid of animals. But there is a world demand for protein, there is a world demand for milk. So the challenge is not to reduce emissions, but it's to reduce emissions cost effectively while supplying the world with food. Methane is influenced by the quantity and quality of the feed an animal eats. So can we change the feed? Can you get feeds that produce less methane?
Achieving even a small reduction in the methane emitted by animals could have a huge effect on global emissions and turn New Zealand into a world leader.
The only real test of whether you reduce methane is in an animal, is you feed it to the animal and does it reduce methane. The problem is that that's a slow and quite an expensive method of doing it. So what we have tried to do are develop facilities that allow us to rapidly screen large numbers of plants.
The testing process that has been set up is overseen by Gerald Cosgrove. How many plants are you looking at?
We've started off, we've got 100 plants on our list to screen.
And that's just the beginning.
Absolutely.
We have what we call batch facilities where we can, in a very simple test tube test, check whether they reduce methane for a short period of time, 24 hours.
Methane is produced in the stomach or rumen of animals such as cows and sheep, and then burped out. So the test facilities try to mimic this environment.
And each one of these has the rumen fluid and the plant in it itself.
A sample of a plant, yes, and that's simulating fermentation that normally takes place in the rumen of a
So this is
sheep or
almost like
cow.
a small replication of a
It's
cow's rumen.
as close as we can get a mini rumen.
So what are you measuring in here?
The gases being released from those fermentation bottles is fed from the bottle through the tube into this machine here, gas chromatograph, which measures the concentration of methane and hydrogen.
If they look promising, we can then take them to another facility, which is, it's an artificial stomach, where we can look at whether that short-term reduction continues for a longer period and in a more realistic situation. This is when we can keep the microbes that reside in a rumen stomach actually alive for about a month.
Right, so what's this machine?
Inside there's a larger bottle where the fermentation is taking place. It's larger than what we saw previously.
It's a fairly accurate representation of a cow.
This time, the artificial rumen are larger and automatically fed for a longer period with the plant that is being tested.
So what are the outcomes you're hoping from this process?
If it's promising at this stage, then we have a greater degree of confidence that that's a plant with real potential.
And then if they pass that test, we can put them in an animal. So this gives us the ability to screen large number of plants in a cost-effective and rapid way. It is a great challenge. It's a great challenge for mankind in general. And I think, I'm extremely optimistic that we can do it. We shouldn't be looking for very short-term solutions here. I think we're looking at a series of small solutions that become additive rather than a simple big win that we can point to and shout eureka.
One of these small solutions has been found just across the road, where a scientist has already made a methane-reducing breakthrough with one of our most abundant and resilient plants. Kerry Hancock has made an amazing advance for New Zealand science by genetically engineering our common clover, the white clover. This plant thrives in our pastures. Kerry's achievement was to make it produce chemicals called condensed tannins.
One of the effects of this is that it decreases the methane that the cow produces.
In fact, methane could be reduced by up to 15%.
Scientists have been trying for 50 years or more to try to introduce tannin into white clover. And so I decided, well, I'd step right back and say, what's another way of doing it that's very, very different?
The only clue that Kerry had was another species of clover called rabbitfoot. This species does produce tannin in its leaves, but doesn't grow well in New Zealand pastures.
So my question was, how come this species can do it, but white clover cannot?
Kerry began the painstaking process of sifting through rabbitfoot's tens of thousands of genes. This would be a big enough challenge, but Kerry had another one to face. The project wasn't funded, so Kerry did the research in her spare time.
I spent some nights out here, my technician as well, and we just slowly, over 5 years, progressed this interest on the side, and it paid off. We'd narrowed it down to about 200 genes that might be responsible for making tannin. Then we narrowed it down to even more, about 20, and then we took a gamble, taking an educated guess that we thought we found the one that was responsible for making tannin.
Kerry finally introduced the gene into white clover, but the most nerve-wracking moment was still to come, staining the leaves to test for tannin.
The most exciting thing was when we finally stained the leaves and they were positive. I was so, couldn't really believe it was real, so I made my technician do it twice because I was, it's such a holy grail to actually solve that I really needed to know before I went and told my boss that actually we'd cracked it. I was really wrapped. I got a bottle of bubbly and a tiny Lamborghini. That's my reward.
New Zealand has some of the strictest regulations regarding genetically engineered plants in the world, so the research is now heading overseas for field trials.
What I'd really like to see is the white clover out in the field producing levels of tannin that will help to reduce the methane. I believe it's one of the methods that could be used to reduce the methane produced in New Zealand.
Our scientists are working hard to try and understand and answer the climate change question. But it's a global problem, so it's going to take a global effort to find the solution. See you next time.